Hello, my name is Batuel, Okda, and I'll be presenting over the complement regulatory proteins and their role in HCV-related pathogenesis. My mentor is Dr. Ross McCall. A little bit of background information. Hepatitis C virus, or HCV, is a positive single-stranded RNA virus, and about 80% of the untreated HCV liver infections will result in a chronic inflammation. About 20% of those individuals with chronic inflammation will develop the scarring of the liver called cirrhosis. 5% of the individuals with cirrhosis will develop hepatocellular carcinoma or HCC. Our complement system is a crucial component of our innate immune system, and it plays a vital role in the antimicrobial defense. Our complement regulatory proteins, CRPs, CD46, CD55, and CD59, are expressed during homeostasis on our cells to protect us against our complement mediated injury. But however, they are overexpressed in many tumor cells, which ultimately leads to be unfavorable. The HCV viral proteins have been shown to regulate the CRPs CD55 and CD59 on hepatocytes during inflammation. However, the role of CRPs in the development of the HCV-mediated cirrhosis and the overall transition to HCC is still poorly understood. As you see in this figure, we see the chronic hepatitis, which is a chronic inflammation of the liver, will result in cirrhosis, which is the scarring of the liver, which ultimately leads to hepatocellular carcinoma with cirrhosis. In our current study, we're going to look at the mRNA expressions of the CRPs CD46, CD55, and CD59 by looking at normal human liver tissue, HCV mediated cirrhosis, and HCC liver tissues. De-identified and discarded normal HCV-mediated cirrhosis and HCC liver tissues were obtained from NIH liver tissue and cell distribution system located at the University of Minnesota, Minneapolis. In order to observe the mRNA expression, we will use real-time quantitative PCR. As you see, there was a non-significant decrease from normal to cirrhosis in the CD46 mRNA expression. And then in the CD55 mRNA expression, we saw a non-significant decrease from the normal to cirrhosis. But however, in the CD59 mRNA expression, we saw a significant increase from normal to cirrhosis and a non-significant increase from normal to HCC. CD46 and CD55 and CD59 all have a very unique role in the inactivation of the complement system. So we observe the ratios to see the correlation or the cooperation together they have in the HCV-mediated cirrhosis and HCC. As we look at the ratio of CD46 to CD55, we don't see a significant change or non-significant change between the disease groups cirrhosis and HCC compared to normal. Whenever we look at CD46 to CD59 mRNA expression, we see a non-significant decrease from normal to cirrhosis and a non-significant decrease from normal to HCC. When we look at the ratio of CD55 to CD59, we see a significant decrease from normal to cirrhosis and then a non-significant decrease from normal to HCC. So overall, we saw a significant increase in the CD59 mRNA expression and a significant decrease in the CD55 to CD59 mRNA ratios in the HCV-mediated cirrhosis for both. CD55 and CD59 relationship in HCV viral proteins mediated inflammation during cirrhosis development needs further investigation. An increased sample size with an inclusion of both genders will be useful to confirm these observations. Mechanisms involved will be confirmed in an in vitro model of HUH7 cells. CD55 and CD59 may serve as a future prognostic markers for HCV cirrhosis to HCC development. And then there's an acknowledgement for Dr. Ross McCall, Sarah Greber, who did a lot of the prior work, Anna Platt and Magdalene right now are in the lab, and I took a sin for mentorship throughout my master's. Thank you.